From Microbe TV, this is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024, starring Cindy Leifer and Brianne Barker, and featuring Robert Binder from the University of Pittsburgh.
Welcome.
Thank you very much. I'm delighted to be here.
Great. So the way we usually do this is we just ask you a little bit about your history. Like, when did you get excited about science, and especially immunology? And then you can talk a little bit about your graduate school training and postdoc for us.
Okay. I've always been interested in science. Even when I was a little kid, I've always been curious about how things work. It can be a Lego toy. It can be being outside with nature. Trying to figure out how things work in not only in biology, but also in physics and in chemistry. And so I was always good in school with my grades in the sciences. So I think at a very young age, I knew that I was going to be doing something related to science. I grew up in a small country in West Africa called Ghana, and that's where I got my bachelor's degree. And I applied to schools here in the United States to pursue a higher degree. And I got accepted first into Fordham University in New York City. Mm-hmm. And I got accepted into a biological science program. So the program covers a lot of many different disciplines. And as a first-year graduate student, you do rotations in different labs to find your niche. And that's when I got into the lab of my mentor, Dr. Pramod Srivastava, and he was an immunologist. Mm-hmm. He specialized in cancer. And that is when I really got excited about immunology. And I got involved in projects related to how the immune system recognizes cancer and addressing very fundamental questions for my thesis work. And halfway through my graduate program, my professor decided to move to University of Connecticut. Yep. And so being a graduate student, I had one backpack. I moved with him. And so I actually graduated from the University of Connecticut in 2000. And then...
Can you talk a little bit about the research you did as a graduate student? Because I'm actually familiar with this research.
Before that, had you done any research when you were an undergraduate in Ghana? Or sort of how did you get the idea that research and graduate school was what you wanted to do? Okay,
so as part of my undergraduate work, I did a little bit of research. It wasn't anything groundbreaking. The finances to do research are very limited, and you're actually constrained in what you can do. So I did a little bit of experiments, but it wasn't anything worthwhile talking about. But the passion was there. I could see the potential of what research
that is worth talking about.
Uncover, right? So as part of my graduate school work, we're trying to... I'm going
to interrupt you one more time, because how did you then know? I mean, you went halfway around the world.
Yes.
Like, how did you know that you were going to do a PhD? There must have been a mentor in your life that told you that was a possibility.
Yes. I mean, a lot of it came from my passion, right? Of wanting to understand how things work, right? So you can dabble a little bit in one area, dabble a little bit in another area, but you can only make so much, you can only make advances to some level, right? But you can appreciate that there's always more to dig into.
Yeah.
So part of my undergraduate research, we were looking at soybeans and finding out how the different properties of different soybeans. And so the very simple experiments were to grind up these different varieties of soybeans and look at the protein profiles there. But I couldn't do anything much beyond that. So I could tell there were differences. I didn't know what the differences were or how they contributed to nutritional values in those soybeans, for example. Mm-hmm. So I left somewhat frustrated, right?
Yeah.
Because there's more to do, but you actually cannot, pursue that to any further degree. So that's where I left it as far as undergraduate went. And in order to pursue a PhD, I knew Ghana was not the place I could do that. And so I applied to as many schools as I could here. It's very difficult to get into a program here from coming from a place like Ghana. So I applied to, I think, 50 to 60 different schools.
My gosh.
Oh, wow.
Almost every school where they didn't require an application fee, I applied to. And I got accepted to Fordham University. And I was glad I ended up there because that's where I met, my mentor, Dr. Srivastava. He's the one person who actually developed my career and, put that research bug in me. And so as part of my undergraduate work, we were trying to understand how the immune system recognizes cancer cells. And my mentor had uncovered these proteins called the heat shock proteins, which can be released from cancer cells. And we knew they were being recognized by immune cells called the macrophages or dendritic cells. But how that interaction happened, we didn't know. We had some hints that there was a receptor on the surface of these cells that was recognizing these proteins. And my project was to find out what this receptor was. And
I just want to ask you if you can describe a little bit more about, why is it important that these heat shock proteins that are released from cancer could be used by the immune system? Because they have a very important role, right?
Yes.
It's not because they're a conserved host protein. So how does it actually identify a cancer cell?
Correct. So these proteins, they were first discovered back in the 1960s. It was actually accidental. There was a technician in the lab who was walking past the incubator and where they were growing cells. And I think he bumped into the dial for the incubator. In those times, they had dials. And the temperature was bumped up a few degrees. And when they came back the next day, they found the cells growing at these elevated temperatures. They had increased expression of these proteins, and they call them the heat shock proteins. They are intracellular proteins. You're correct. And most of the work on heat shock proteins have studied their role inside cells, how they chaperone things inside the cells. My mentor was the first one to show that when you have a compromised cell, like a cancer cell, some of these proteins can be released into the extracellular space. And that's where they act as a danger signal for the immune system. They actually trigger immune responses. And the first cell to engage these heat shock proteins are the macrophages or the dendritic cells. So in my project, I took the membranes of these macrophages or dendritic cells and then extracted the surface proteins, and see which of those proteins interacted with the heat shock proteins. And right when I was graduating, we found this receptor called CD91, also called LRP1. And so we published a major paper on that protein. And traditionally, when you finish your graduate work, you go on and do a postdoc, which is further training in research. But I just uncovered this protein, and there was so much that was still unknown. So I had a discussion with my mentor, and I decided to stay to do a postdoc in the same lab and to find out what this receptor actually was, how it elicited its function. And so I did a postdoc in the same lab, which is usually frowned upon. But I had a very successful postdoc. I published a lot of papers on that receptor. And towards the tail end of my postdoc, there was a talk in our department by a lady called Dr. Olia Finn, and she was the chair of immunology at the University of Pittsburgh. Yep. And she said they were hiring, new faculty, and I went to interview in Pittsburgh, and I got a position there as an assistant professor, and that's where I've stayed all my career. It's a wonderful environment to do research, and it's a place where my career has really flourished.
That's awesome. Yeah. So can you tell us a little bit about what happens in the macrophage after CD91 senses heat shock protein? Yes. You're talking to two people who think about macrophages and innate immunity, so we're excited.
Okay, yes, I can. Of course. So it's actually still a big part of what I do now. We're working in this space called cancer immunosurveillance. And I usually tell people to forget about most things that you know about big, bulky tumors, which is where a lot of people are basing their research on. When you talk about immunosurveillance, cancer begins with a single cell that has some genetic abnormality, and this cell will begin to divide abnormally until you have a big mass of cells. But very early in the response, when you have the first 10, 50, 100 cells in a tissue that is where they are beginning to divide, you already have an immune response against those cells. You don't have a true tumor microenvironment where things are dysregulated. And so one of the questions I have tried to answer is how the immune system recognizes these very few aberrant cells very early in the tissues. And we think this heat shock protein CD91 pathway is critical in that space. So if you look at individuals who are immune suppressed, people who've transplant patients, for example, who are put on immunosuppressives, or HIV patients, and their incidence of cancer goes through the roof, right? Mm-hmm. It's much higher. And the idea is that our immune system is constantly finding these cancer cells. We've all generated cancer cells in our tissues many times over. And if your immune system is up to speed, then it recognizes these cells and eliminates them. So this is something that is happening. This pathway is happening most often. So we created a mouse where we knocked out CD91 expression in dendritic cells. And if those mice are very similar to immunosuppressed individuals, they develop cancers at a much higher frequency. And so we know this pathway is critical for preventing development of cancer in mice. We've also looked in people, and many cancer patients, they either have a deficiency in CD91 expression, it's lower than normal, or they have a mutation in the receptor which dysregulates the receptor, and so it no longer functions. So coming to your question, how does this all work? So when heat shock proteins bind CD91, CD91 is responsible for two things. The heat shock proteins are chaperones. So they are bound to material from the tumor cell when they are inside the cell. And when this tumor cell releases the heat shock proteins, they are carrying all of this cargo with them to the extracellular space. So CD91 is an endocytic receptor. Heat shock proteins bind, and the heat shock protein is endocytosed into the dendritic cell or macrophage. But it's coming along with all the cargo that it has picked up from the tumor cell. And some of these are the tumor antigens, or the mutated proteins and peptides from the tumor cell. So this material is processed within the macrophage or dendritic cell and is displayed on the cell surface by MHC molecules. So this can be recognized by T cells. And it'll be a T cell response that's specific for whatever the tumor is expressing. But before the T cells can get going, before they're activated or primed, they need other signals like cytokines, which we call signal 3, and they also need co-stimulation, which we call signal 2. So when heat shock proteins bind to CD91, we also know CD91 will signal within the cells. There are lots of gaps that we don't know how CD91 signals inside the cell. It's something that we're actually presenting at this conference here. We've uncovered some signaling molecules that are involved in transmission of the signal. But the net result is that these dendritic cells or macrophages become activated. They begin to release a boatload of cytokines, and they upregulate co-stimulatory molecules, specifically the dendritic cells. Mm-hmm. And so dendritic cells that express CD91 that have engaged HSPs, they express all the signals needed to get T cells up and running. They are primed, and those T cells can find tumor cells and eliminate them. So this is how we believe the pathway goes as far as recognition of the cancer cells and elimination of the cancer cells. And if you disrupt the pathway, either by blocking HSPs from binding CD91 or removing CD91 from the equation, then cancer cells win out and you get disease. These cancer cells just keep proliferating until you have big tumors.
Have you looked at all of whether the cancer cells actively downregulate this to promote their, survival and growth?
That's a beautiful question. Yes, they do. So there is actually a protein that can disrupt the engagement of HSPs with CD91. It's a protein called RAP. And we can find some tumors that express RAP that can be released along with the HSPs, and that will interrupt the the pathway. So this would be an escape mechanism, right? By which tumors can win out.
Are there any cases where these HSPs are released in an infectious setting?
Yeah. Yes, absolutely. Yes. So we believe. So our I don't like to use the word believe because that's more of a faith-based thing. But our idea is that this pathway can happen during infectious disease as well. There are because infectious agents, viruses, bacteria, they undergo very rapid proliferation. There's a boatload of antigens that are available. And so in some situations, they can circumvent this pathway and can use other pathways to transfer material between the infected cell and dendritic cells. So in infectious disease, the HSP CD91 pathway is not always required. There are other pathways to stimulate, T cell responses. Viruses and bacteria will express molecules that can trigger the toll-like receptors, so other receptors besides CD91 that can trigger these signals to get T cells going. Now, having said that, if you there was a study that we were involved with many years ago. There was a cohort of sex workers in Uganda. They were sampled for their HIV positivity. They found, so they're all exposed to HIV on a constant basis. Some of them got HIV, some of them did not get HIV. And when they looked at all the HIV negative patients, individuals, they all had elevated CD91 expression. Right?
Well, that's interesting.
So there's some correlation there. And we have come to the idea that high expression of CD91 correlates with immune fitness. It's an indication of how fit your immune system is.
Okay.
And so you'll be better protected from cancer and most likely infectious disease.
Very cool. I was aware of that cohort of patients, but actually hadn't heard the link to CD91 before. Right. This is awesome.
Yeah. There were 2 papers that were published on that a number of years ago. Now, because we're heavily focused on cancer, we haven't had so it remains a correlation, right?
Right.
And we have to definitely dig down to see
cool.
what the mechanism is. Right? Yeah. So.
What about autoimmunity? Because, I mean, in the cancer cell, there's going to be some mutated proteins, but there's the vast majority of the proteins are just going to be normal host proteins that would be chaperoned by HSPs unless there's some selectivity. So how do you not break tolerance, or is there a role potentially for this pathway in development of autoimmune disease as well?
Yeah, beautiful question. So in as far as the self proteins go, and self-peptides, they are chaperoned by the HSP. So when you get transfer, majority of what is being transferred are going to be self-normal proteins, normal peptides. And they will all be processed by dendritic cells or macrophages and displayed on the cell surface by MHC molecules. However, we don't have T cells that recognize those cognate antigens.
Hopefully.
Right. No, because they're eliminated by during development in the thymus, right? Now, having said that, I would not rule out the fact that you could potentially trigger, a self-reactive response. Now, a big part of this story, and it also addresses the autoimmune question. A big part of this story is that we have been using HSPs as cancer vaccines, right? Because if you take a cancer cell and you take it to a lab, you grind it up, and you purify the heat shock proteins from the cell. Now you have the HSPs with all the antigens that are being synthesized in the cancer cell. So you can actually vaccinate somebody with these heat shock protein preparations. Now, if you have a tumor, let's say on your left arm, we'll vaccinate on your right arm where you have healthy dendritic cells. When you inject the heat shock protein, you're putting it in the extracellular space. So this should be picked up by normal functioning dendritic cells, and they should prime a good T cell response. So there were a couple of clinical trials that were run years ago. They did show efficacy that you could actually prime T cell responses. And we've been doing this in mice for many years. with different types of tumors, different strains of mice. So we've done that in mice. We've done that in people. There have been other labs that have done this in rats. I think there was a small study in dogs as well. And in no case have we vaccinated with HSPs and seen any autoimmune.
That's amazing.
Reactivity,
Wow.
right? And even though we know that what we are vaccinated with, majority of the peptides or proteins associated with HSPs are actually self. It's going to be a very, very small, less than 1% of those, of the cargo is going to actually be tumor antigens that are specific for that tumor. 99% or even more is going to be self-normal, proteins. And people have looked at that. You can actually strip off the cargo from the HSP and do a mass spec, for example. Most of it is just normal.
Wow.
Wow.
But you never
go tolerance.
Right. Yes, go tolerance, right? Absolutely. Now, the other way to address this question is, so we've studied animals where we've removed CD91 from the equation by knocking it out, right? Or in people where they have downregulation of the receptor. Now, if you want to go the other route and put it back, how much CD91 do you want to put back in the mouse, for example? And is there the idea that if you put back too much CD91,
could you then break tolerance?
Break tolerance. So yes, so in people where they have a downregulation of CD91, yes, we want to fix that, right? So if you have a way to reverse that, it would be wonderful. But we have to be careful how much you put back, right? In order not to trigger. So we'll do this first in animals and see how it works, and then we'll think about people.
That's really interesting. I want to switch gears a little bit because I know you do some teaching too, and you have a passion to work with early career scientists. So can you talk a little bit about that?
Absolutely. So I would characterize early career scientists as young as you could possibly go. Because I
the kids playing with the Legos like you did. I like it.
Absolutely. Because it starts at a very young age to develop that passion. And, every year I've been going to local high schools in Pittsburgh. And I spend about an hour with, middle school kids and with high school kids. And I tell them very simply what it's like to be a scientist, what my day-to-day activities are like, and how great a career in science is. And at that age, they're thinking more about NFL. They're thinking about, maybe I'll become a doctor, but I'm not sure yet. But just telling them, how wonderful science is. Yeah. And one of the experiments that I share with middle school students, for example, is just looking at GFP, for example, discovered in these sea creatures, and how you can tag things with GFP and light up organisms and tissues. So I told them about viruses. I asked them when you transmit COVID, for example, from one person to the other, where does it go? They say the lungs. I'm like, wonderful. How do we know it goes to the lungs, and how can we explore that? And so I share with them, you can actually put GFP on a virus, and you can follow it and light up lungs of organisms. And they, something as simple as that, they get very passionate about it, and I get lots of thank you notes, and I make sure I respond to them. And we've had students over the years follow a career they're now in undergrad, pursuing a science career. So catch them young, expose them to how wonderful science is, and many of those will end up becoming the next generation of scientists and doctors.
That's fantastic. Yes. I'm so glad that you have that passion for it, as that's so important for all of us in the sciences.
So do you have any mentoring advice for trainees? Maybe not high. Well, I mean, you could talk high school students. They're probably watching this, but there's a lot of trainees that watch our show. Do you have recommendations?
Yes,
for
absolutely.
Careers in science?
Yeah, absolutely. So from a mentor's perspective, I always try to personalize my mentoring style, because every student is going to be different, and they all require different things as far as their progression through their programs, through their studies. And so I at the very onset, I make sure I understand the student and make sure I know what they need as far as their development goes. And the reverse is also true. They, the mentees need to pick a mentor who can understand them, who can develop a relationship with them, and provide them with what they need as far as what their weak points are, and enhancing their strong points. So that relationship is very, very important, and nothing should be glossed over. If very early on the mentee sees something in the mentor that makes them stay up at night, it should be addressed, or maybe find a new mentor. Right? Yeah. There are lots of them around, so it's a very important decision. It can be a deal breaker as to whether that mentee goes on and has a successful career or not.
All right. So I have a slightly different question that we often ask on our other podcast, TWIV. So if you hadn't been a scientist, what do you think you would have become?
That's a very difficult question. I never really had a backup plan. This might be scary, but yeah. It's I never really had a backup plan. Maybe it would not have been immunology. Maybe it would have been another science area of investigation. But it would have been something to do with science making discoveries. I think that
shows your passion for science.
Yeah,
makes a lot of sense.
Yeah.
Thank you for joining us.
Thank you very much for having me.
That's a special episode of Immune, an immune booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today is from the University of Pittsburgh, Rob Binder. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, the podcast that's infectious.